work with very, very excitable things. And I'd like, well, got it. Yeah, I'd like to say some words about Dr. Athma Pai, just to share my screen a little bit here. Thiago, você pode deixar eu compartilhar minha tela?
Ok.
I need to... Obrigada, Thiago. Not yet. Okay. Here. Well, Dr. Athma Pai is from the RNA Therapeutic Institute from University of Massachusetts Medical School, United States. And she's going to talk about the efficiency of gene regulation, how speed and accuracy influence mRNA processing. He's going to tell us about a little bit of her group is studying now, and they study the speed and efficiency at which RNA molecules are created and processed to ensure proper cellular functions, combining high-dimensional computational analysis with state-of-the-art genetic, molecular, and cellular genomics techniques to probe mechanisms of RNA maturation. She has 2 lines of research: kinetics of mRNA processing and dynamics of RNA processing. Both lines of research are very important for us, especially my group. I work with alternative splicing, and then I'm very excited and very anxious to listen to you and to know the state of the art in dynamics of RNA processing and kinetics of RNA synthesis. Thank you very much, Dr. Athma, to be here and to give the seminar in this BRAZWEBINARs from our PhD program. Not PhD, it's a graduate program. Thank you very much. We really appreciate it.
Great. Well, thank you to Gloria and to Miguel for this opportunity, and to Lisa for suggesting my name. I think this is a really great opportunity during the middle of the pandemic to still see and meet new people, which I'm very excited about. So let me just make sure that this works here. Does this work? Is this okay? If somebody can just indicate that this is fine. And
you're just not on presentation mode.
I'm not on presentation mode?
Not on the presentation, just showing your slides.
I was wondering about that. Okay, give me a second. I gotta change. Sorry, I'm usually not at work these days, so I had to change this a little bit. All right, let's try this. Do the desktop. And all right, how about now?
Yeah,
great.
That worked.
Excellent.
Great.
Okay, sorry about the delay here. It's shocking how after a year and a half of Zoom, it still takes a while to make things work the way you want them to. Okay, well, it's my pleasure to give a talk today to all of you. As Gloria mentioned, I'm going to talk on a topic that's really central to my lab and give you a flavor for some of the projects in the lab relating to this overarching question of how efficiently do cells create mRNA, and how specifically do they balance the speed and accuracy of the underlying mechanisms in order to regulate gene expression. I'm happy for anybody to ask me questions in the middle of the talk or afterwards, so please feel free to interrupt if you'd like. Okay, so as we're all very familiar with, this is the central dogma of molecular biology, where RNA is transcribed from DNA molecules, and that RNA is then, sorry, now just want there we go, okay. That RNA is then translated into protein molecules. However, this depiction is really an oversimplification of the underlying processes that underlie any of either transcription or translational processes, because they show them as this magical one-step process of transcribing RNA from DNA or protein, translating protein from RNA. However, in reality, each of these processes are actually comprised of many individual molecular mechanisms that act spatiotemporally in a coordinated spatiotemporal fashion to actually to create RNA or protein molecules. And so these include mechanisms including not just transcription, but also downstream processing, including mRNA splicing, 3' end processing, mechanisms involved in export, subcellular localization, and a whole host of other mechanisms involved in translation. So one of the central theses of my lab is that each of these individual molecular steps actually takes a certain amount of time and is coordinated in a certain fashion. We know a lot about the individual mechanisms involved in each of these steps and the individual alternative decisions involved in creating a final transcriptome. We actually know quite a bit less about how they're coordinated. And one of the main thrusts of my lab is that in order to really understand how they're coordinated, we actually also have to understand the timescale at which any of these processes occurs, and therefore the aggregate timescale at which an RNA or protein molecule is created. So for instance, for RNA molecules, the time spent in any one of these steps might actually influence the final isoform that's generated from the same genetic sequence. So perhaps for certain genes, spending a lot more time transcribing that molecule might make downstream processes go quite fast and generate a certain isoform. And for the same gene, if the cleavage step, for example, takes a long time, it might lead to the generation of a different isoform from the same genetic sequence. So these, each of these individual mechanisms and the dynamics of decisions involved in RNA processing are actually quite important. I'm simplifying the processes up here into three main overarching mechanisms that we work on, including the initiation and elongation of transcription, the splicing of mRNA, and the cleavage and polyadenylation that generates the 3' end of the molecule and influences 3' UTRs. What we know, and this is from a review that we published a few years ago, is that each of these steps are widely alternatively used across different isoforms from the same gene, and those alternative isoforms are regulated in a context, environmental, or tissue-specific manner. So what I'm showing here is, for instance, that upon chemical stimulation, there's a large amount of differences in transcription, especially alternative usage of transcription start sites. But in contrast, upon immune stimulation, we and other groups have found a lot of alternative exon usage and changes in 3' UTR composition driven by alternative cleavage site usage, and so on and so forth, including differences upon heat shock, DNA damage, cancer, and many other diseases in cellular contexts that I'm not listing here. One of the hypotheses has been that in many of these contexts, the time it takes to generate mRNA molecules in order to respond to an environmental or a pathogenic or an external threat for a particular cell really matters. And therefore, the time it takes to not only transcribe the mRNA, but also process the mRNA might influence the specific alternative isoforms that are actually generated and globally generated upon a perturbation or stimulation. So with that, the goal of my lab is to understand the molecular basis for transcriptome diversity. And we do this by developing and applying genome-wide methods for measuring the efficiency of gene regulatory mechanisms. And I want to highlight that the way in which we do this is by using both experimental genomics and computational biology tools. Our lab does about 50-50 of each of these, where I have students and postdocs who generate the data and then are able to actually analyze it within the context of their project. So we really aim for an integrated lab where we're really focused on the questions rather than only the techniques that we're using. So the primary question that we're starting to tackle is, what are the rate limiting steps for RNA maturation? And so what do I mean by this? What I mean is that, again, in order to create an mRNA molecule, you have to undergo a number of steps, simplified here to transcription, splicing, and 3' end cleavage. And the time it takes, so for certain sets of genes, the time it takes to transcribe the molecule might actually be the rate limiting step to create that final isoform or that final mRNA molecule. However, for other genes, the time it takes to splice or the time it takes to cleave that molecule might actually gate the production of a final mRNA molecule, while transcription of the necessary sites occurs relatively fast. So in aggregate, understanding which of these specific steps is actually limiting the production of an mRNA molecule would allow us to understand the efficiency and the mechanisms that underlie alternative splicing, alternative cleavage, and how a cell generates the proper transcriptome that it's needed to either survive a external attack or to happily grow in any condition. So the first process that really started me on this venture was studying mRNA splicing, as Gloria mentioned, and my background was really in understanding the kinetics and dynamics of mRNA processing. So the first story I'll tell you about today is measuring the rate of mRNA splicing. So splicing, as many of you probably know, is an essential step in the mRNA life cycle, and it involves excising an intronic sequence from a pre-mRNA molecule and joining two exonic sequences to form a mature mRNA molecule. And so the process to do this uses specific sequences within the pre-mRNA molecule, and specifically within the intron, including a 5' splice site, a branch point, and a 3' splice site, and involves two catalytic steps to actually to remove this intron and result in this spliced molecule and an intron lariat that gets degraded quite fast and does not, we think, influence the final transcriptome or the final protein products produced in the cell. So mRNA splicing is also very important for disease and many other cellular contexts. And so mRNA splicing has been known to be dysregulated, or the pathways involved in regulating mRNA splicing have been known to be mutated in spinal muscular atrophy, muscular dystrophies, autism spectrum disorders, multiple cancer subtypes, and also involved in immune disease and neurodegenerative diseases as well. So as I mentioned, our main question is how can we actually study the rate at which mRNA splicing occurs? And so the rate at which this intronic molecule is actually removed from a pre-mRNA molecule. And so our first challenge is to actually experimentally identify intermediate RNA products. And so when any of us conduct a standard RNA sequencing experiment, what we usually are doing is capturing only or mostly mature, stable mRNAs that have a cap and a poly A tail, but are not informative of the history that it took to actually generate that mature mRNA molecule. So instead, in order to study the rate, what we want to do is capture nascent RNA molecules before their maturation and degradation of any inaccurate processes that are occurring. So to do this, my lab's main technique is to use a technique called metabolic labeling or 4SU labeling, where we use a synthetic nucleoside called 4-thiouridine, depicted here by this little yellow dot, where we can incubate cells in cell culture with this 4SU molecule for a short amount of time. We can then extract the RNA and use a biotin-streptavidin interaction to specifically biotinylate the 4SU molecules and use streptavidin to pull out any biotinylated molecule. What this allows us to do is allows us to separate the labeled fraction of RNA, which specifically represents the nascent RNA that's been created within an experimentally determined time frame, from the older unlabeled fraction of RNA that mostly represents stable, mature mRNA molecules. Oops. So we do this with very short 4SU labeling time periods, 5, 10, or 20 minutes. And this is different from what many labs studying transcription have done, which usually involves longer labeling periods, such as 30 minutes or an hour, or pulses followed by chases in order to measure mRNA stability, for instance. Using these short labeling time periods to capture RNA molecules allows us to take this nascent fraction and use Illumina high throughput sequencing to sequence millions of short reads from this nascent fraction of RNA. And the way in which we do this is we actually go a step further and we do a time course of this 4SU labeling. And this allows us to capture intermediates at different times after generation of those molecules. So the data I'm going to tell you about today is from 5 minutes, 10 minutes, and 20 minutes of incubation with 4SU. And the idea is that with increasing labeling time, there's increasing time for those molecules to get spliced. And so you can see that more introns are present in the 5-minute time period than in a longer 20-minute time period. We can then model the rate of intron loss to infer the splicing rate or the splicing half-life, which is the time it takes to splice introns from half of the molecules from a particular gene. And we can model the rate of this loss over time to infer the splicing rate or half-life. And as I mentioned, the data I'm going to tell you about today is from a 5, 10, and 20-minute incubation with 4SU, specifically in Drosophila melanogaster S2 cells, which is a cell culture system representing embryonic Drosophila cells. For these data, we have 3 replicates per time point, and we're also paired with a total RNA sequencing data set that mostly represents steady state RNA. And this is actually data that was generated by Karen Edelman's lab when she was at NIHS, and she's now at Harvard. Okay. So one of the main parts to, this is a computational biology or a bioinformatics lecture series, so I really have to highlight why what I'm showing you here with this modeling is actually a huge oversimplification. So one of the main challenges we had is that we needed to model the data and therefore model the splicing rates in a more refined manner because of the way in which the data was generated. So the plot that I showed you on the previous slide makes a big assumption, and that makes an assumption that the label was added exactly at time 0 and that the entire labeling period was available in order to splice out that particular intron. So if this is a 5-minute labeling period, if this intron was created exactly at time 0, and we ask what proportion is left over 5 minutes, then that would be fine. However, the data was actually generated with a continuous labeling over this entire 5-minute or 10-minute or 20-minute labeling period, which actually means that many of the molecules or many of the reads are derived from molecules where the intron was actually transcribed sometime in between the 0 and 5-minute time point. So it includes a mix of reads coming from molecules looking like this, which had a full 5 minutes to be spliced, and molecules looking like this, in which the intron was completed transcription only a few seconds or minutes before the end of the labeling period. Furthermore, we also include molecules where the intron could have actually been completed transcription previous to the addition of the label, but because we're pulling down on the 3' end of that molecule, we'd still see reads like that. And finally, for very, very long introns, transcription could still be actually actively ongoing within those introns and therefore entirely throw off our time periods. So if we knew which reads came from which of these time periods, we'd actually be able to go in and ask for each of these intron lifetimes and each of these introns, what proportion of reads do we see that are intron-exon junction reads, indicative of splicing, of unspliced molecules, or exon-exon junction reads that bridge the 2 exons and only occur after this intron's been spliced out, and therefore indicative of spliced molecules. So if we knew which of these reads came from each of these lifetimes, we could easily model this, but of course we don't. And so what we have is instead an amalgamation of all of these lifetimes into one giant pool of reads. So the challenge here was really to try to address how do we infer the distribution of lifetimes using a population level data set. And so what we decided to do was we decided to specifically model the distribution of polymerases that could actually give us this, this distribution of lifetimes in our data. So I'm not really going to go into the math here because this paper is actually published. But essentially what we're doing is we're saying that given a uniform distribution of polymerases across a region that could provide us with molecules given our labeling time point, we can model the probability of observing unspliced reads, which decreases over time, and the probability of observing spliced reads given a particular half-life. So it turns out these two measurements are actually the count of unspliced and spliced reads is what we have. And so these are the empirical measurements of intron-exon junction reads and exon-exon junction reads. And we can take the ratio of these two and model these probabilities and solve for H, which is the half-life in this particular equation. So we do this. So before I show you the results from this particular model, I want to show you what some of the raw data actually looks like. So these are read coverage plots for 2 different introns in the Drosophila genome. And so on the top here, I'm showing an intron that's 63 nucleotides long and has a half-life of 1.31 minutes. And what you can actually see is that the purple reads are intronic reads, the gray reads are exonic reads. And what you can see is that over time, you see fewer intronic reads relative to a more stable or constant count of exonic reads. And when we pull out the junction reads that were specifically informative and that we're using for our modeling data, the blue are exon-exon junction reads, which increase over time, and the pink are intron-exon junction reads, which very reassuringly decrease over time. And so we can do this for each intron. On the bottom, I'm showing you a much longer intron where you can see many of the same patterns. So we can do this for every single intron that we have sufficient read coverage for in our data set and actually calculate the half-lives. We're able to calculate half-lives for a little bit more than 25,000 introns in about 5,600 genes in the Drosophila genome. And we arrive at a median splicing half-life of 1.96 minutes. So the first question we wanted to ask, because we wanted to be able to use this method to actually infer something about the mechanistic biology underlying splicing, was what actually underlies variability in splicing rates. And so one of the first things we wanted to ask about is, does the length of an intron actually influence the rate at which it's spliced? And this has actually been hypothesized to be the case through a model called a diffusion model, where the length between the 5' splice site in blue and the branch point in yellow, which is the first catalytic step of splicing, the joining of these two sites, might actually influence the longer the length, the longer the time it might take through diffusion for these two sites to come together and catalyze splicing. So what I'm showing you here is a plot showing the correlation between intron length on the x-axis and splicing rate as modeled by half-lives on the y-axis. And each of these dots is one of these 25,000 introns we were able to measure splicing half-lives for. In blue is a running median of the intron lengths, of the half-lives over the intron length distribution. And what you can see is that in general, there's no or very little correlation between intron length and half-life. Is there a question? Maybe not. So one thing stands out, though, when you look at this distribution is that while over the entire distribution, there's very little correlation, there's a huge dip right around 60 to 70 nucleotides. And that was pretty intriguing, given that this is in Drosophila cells. And one thing that's known in Drosophila cells is the modal length of a Drosophila intron is 63 nucleotides. And greater than 30% of introns lie within 60 to 70 nucleotides long. And this is due to selective pressures in the Drosophila melanogaster and other Drosophila genomes influencing or favoring shorter introns. So when we zoom in right into that much shorter length range here between 40 to 100 nucleotides, we really see this dip, and see that this dip is very specifically at this modal intron range of 60 to 70 nucleotides, where when you go slightly longer, you get slightly slower half-lives. And also when you go slightly shorter, you get slightly slower half-lives. And so it seems there's an interplay between the length of an intron and the optimal rate of splicing, such that the modal length of an intron seems to also match up with the fastest splicing rate. And so it's a very interesting situation in which we think there's selective pressures on optimal splicing that are competing with or collaborating with, in some ways, the selective pressures favoring shorter introns in the Drosophila genome, setting a limit to how short you can really go before hindering the splicing reaction. And so, so this begs the question, why can't you go any shorter? Why can't you actually go to 40 nucleotides? Wouldn't that actually be better for not only the diffusion between different sites to create more efficient splicing, but also for shorter introns in the genome? And so to ask this question, we actually went in and asked about what are the positioning of the various types of sites in the Drosophila genome and the strengths of either the 5' splice site, branch site, or 3' splice site. So what I'm showing here is a distribution of splice site scores is modeled by a maximum entropy model across different intron length ranges. And what you can see is that while 5' splice site scores are relatively the same across intron lengths, there's a drastic reduction in 3' splice site strengths or scores in this very short intron range. And when we went looking why that might be and included the position of the branch site, we see that for very short intron lengths, the branch site is actually very close to the 3' splice site. Versus for longer intron lengths, the branch site is about between 25 to 30 nucleotides away from the 3' splice site. And so what this implies is that for very short introns, between 40 to about 60 nucleotides long, there's probably not enough room to have both the proper branch site and a proper polypyrimidine track upstream of the 3' splice site, as is necessary for recognition and proper splicing to occur. So this is quite interesting because it marries the biochemical and genome architecture features that evolution has to act upon to optimize both mechanism and other selective pressures influencing the genome. Okay. So finally, one of the last things we were interested in is whether or not there was any influence of alternative splicing on the rates of different introns that were being spliced out. And so while in our model, we could only really model the rates of constitutively spliced introns, we could ask about whether or not an intron that was alternatively used across cell types could actually have different rates within a cell type where we knew what the usage was. So what we did is we broke down our introns into constitutively used introns in which the intron was always constitutively spliced out, regardless of cell type, given all of the Drosophila data that we have in the genomics literature these days, or whether or not that intron was annotated as being either retained or alternatively used around a cassette exon in other cell types. So retained intron would be an intron that was actually regulated to be included in the final transcript, and a cassette exon flanking intron was an intron that flanks a skipped exon, where the skipped exon could either be included or excluded in a final transcript. And so what we see is that these two classes of alternatively used introns actually had slower half-lives regardless of the intron length, indicating that despite the fact that they all resulted in efficient splicing or complete splicing in the Drosophila S2 cells, there was more time taken to actually undergo that splicing decision when there was a possibility of regulating that intron in an alternative fashion. Okay, oops, let's skip this. So that led us to some very intriguing conclusions about how we could actually use splicing rates to understand something about the mechanisms underlie splicing. And so, as I mentioned, what we ultimately wanted to do was to marry this with the rates of other mRNA processing or transcriptional processes that might tell us about the mechanisms underlying overall alternative mRNA usage in cells. So we have a method that we're working on right now to measure the rates of transcription elongation, and that's actually in collaboration with Zhiping Wang, who I think spoke at the same series a few weeks ago. And so next time I hope to be able to tell you about that model. But today I'm gonna continue by telling you about a model where we used a very similar technique to model the rates of 3' end cleavage. And this is work by my graduate student, Leslie. So, okay, so what we were interested in is the temporal control of 3' end pre-mRNA cleavage and polyadenylation. And so we're switching gears a little bit from thinking about an intron and how fast that intron is spliced out in the middle of a transcript to how fast the end of a transcript actually gets determined. And so not only is that end of the transcript and the cleavage and polyadenylation of a transcript really important for generating a mature mRNA molecule that can actually leave the nucleus and be transported to the cytoplasm, but it's also involved in alternative decisions involved in many downstream mRNA processes, because alternative 3' end cleavage can result in different 3' UTRs, which can then be bound by RNA binding proteins, microRNAs, or lincRNAs to influence many processes, including mRNA stability, mRNA translation, nuclear export, localization, and protein localization. So we wanted to know whether or not a measurement of the rates of 3' end cleavage might actually tell us something about how these particular alternative sites were regulated. So furthermore, there's actually been hypotheses in the literature about how various molecular rates might actually influence alternative usage of different types of sites. And so one hypothesis is that the rate of transcription elongation across a gene, and in this case across a 3' UTR, might lead to differences in poly-A site or cleavage site usage, where where a very slow polymerase elongating across a gene might have more time seeing only the upstream site 1 before and therefore cleave before it even reaches the second site, while a polymerase that elongates quite fast might have more time where both sites are present and be more inclined to use this distal or downstream site. However, this hypothesis has another piece to it, because not only does the rate of elongation determine how long these sites are actually present and able to be cleaved, but the intrinsic rate of cleavage at each of these sites interplays with this elongation rate, where this upstream site might actually intrinsically have a faster cleavage rate or a slower cleavage rate, and therefore influence the resulting 3' UTR isoforms. Okay, so these kinetic parameters influencing the selection of 3' end sites have been hypothesized for quite some time, but there's actually never been a method to specifically measure at a genome-wide level the rates of 3' end cleavage and polyadenylation. And one of the reasons for that is likely that it's actually not that easy to figure out how to differentiate between cleaved and uncleaved transcripts using genome-wide RNA sequencing data. And that's because when you have an RNA sequencing data set, you actually have a range of different molecules within that RNA sequencing data set. You have reads from molecules that have not yet even reached the cleavage site and therefore cannot be cleaved. But among the molecules that have actually transcribed past this cleavage site, you have both cleaved and uncleaved molecules. And it's very hard to actually differentiate between these two sets because there are no junction reads as we would normally, as we used to model the rate of the rate of splicing in the previous story that I told you about. So when a polymerase transcribes past the cleavage site and cleavage actually occurs, this results in two different molecules: the mature mRNA molecule in dark purple and a read-through region or a read-through molecule in light purple. You might ask, well, why can't we just use the measurement of this read-through region as a way of inferring whether or not that molecule has been cleaved or not cleaved? And we can't, because that read-through region is actually fairly quickly degraded in a model called a torpedo model, where XRN2 comes and starts chewing from the 5' end of this read-through region, and therefore degrades this region quite quickly. So what we're left with is basically mature mRNA and uncleaved transcripts, and it's hard to differentiate between the two of them. So in order to really understand how we might which regions or types of reads we might use in our RNA sequencing data to actually differentiate between cleaved and uncleaved molecules, and therefore ask about the rate of cleavage, we simulated nascent RNA sequencing data in silico using a range of different parameters and asked, well, what patterns do we start to see around the cleavage site? So what I'm showing you here are each row, there's thousands of rows here, but each row is a different pre-mRNA that's been simulated using these parameters here, a particular gene length, a particular read-through region length, expression level, and a number of transcripts or coverage level. The dotted line is the cleavage site. Upstream of that cleavage site is the gene body, downstream is the read-through region. And we're simulating 3 nascent RNA time points, similar to the data that we actually have in Drosophila. So we can then actually simulate the rate of cleavage. And what I'm showing you here is a 5-minute cleavage half-life. And what this will do is it'll take each molecule and say, well, if it's transcribed past the cleavage site, with a certain probability, given this half-life, it'll be cleaved or uncleaved. So the gray molecules are molecules that have not yet reached this cleavage site. The black molecules are molecules that have transcribed past the cleavage site but are not yet cleaved. And both versions of purple molecules are molecules that have transcribed past the cleavage site and are cleaved, and therefore there's either a mature mRNA and both a mature mRNA and a read-through region for these molecules. We can then undergo the actual process in silico of creating an mRNA sequencing library from these nascent RNA sequencing data. So we can conduct random fragmentation. We can undergo, sorry, this takes a minute. We can undergo size selection with a particular insert size and standard deviation. And what we start to see when we actually undergo this process of creating an RNA sequencing library from these simulated reads, we start to see that there's a depletion of fragments and reads right around this actual cleavage site, where the black molecules that have not yet been cleaved still show read coverage right across this cleavage site. But all of these purple molecules that have been cleaved start to actually lose reads. You see a depletion of reads right around this cleavage site. And so this is actually fairly informative. And when you actually look in aggregate across all of these reads in a metagene plot, you see a very prominent dip of read coverage right at the cleavage site. And we realized that this is actually due to something called an RNA-seq edge effect, where in the process of creating an RNA sequencing library, the random fragmentation and the size selection steps actually lead to a depletion of reads right at the very edges of molecules, due to the fact that the edges of these molecules are likely to have very long or very short fragments that are not likely to be maintained through the size selection step. So what's happening here is that for the proportion of molecules that have actually been cleaved, there are now actually 2 molecules, the mature mRNA fragment and the read-through region, and both of them are subject to edge effects, not only at the very beginning of the gene and the end of the gene, but also right in the middle where they've now been cleaved apart. So because of this edge effect, because there's 2 different molecules, you actually see a depletion of reads specifically for those molecules that have actually been cleaved. Now this allows us to say any read that overlaps the cleavage site is indicative of an uncleaved RNA molecule, but the depth of this particular dip is now actually informative about the number of cleaved molecules that we can see in our data. So this actually taught us a lot about how we might use our data and take advantage of the technical processes of preparing an RNA sequencing library in order to estimate the number of uncleaved and cleaved molecules that we needed across time to model the rate of cleavage. So furthermore, with increasing labeling time points, this dip should actually be more pronounced because there would be more cleaved molecules with increasing 4S2 labeling time points. And what I'm showing you on the right-hand side is a metagene plot across 11,000 annotated cleavage or transcription end sites where we see exactly this pattern. So this is now no longer simulated data. This is real data centered around this cleavage site. And we see this very prominent dip right around the cleavage site. And we see exactly what we observe, that this dip is more pronounced for longer labeling periods and the most pronounced for this total RNA sequencing library that is representative of mostly steady state RNA. Okay. This is not just true in aggregate. We can actually see this for individual cleavage sites. And I'm showing you 2 examples of individual cleavage sites here. And we can then now use these data to actually model the rate of cleavage. We make 3 main assumptions. I'm again not going to go into the math, but kind of give you a conceptual understanding. We make 3 main assumptions about these data. The first is that the polymerase has to have actually transcribed past the cleavage site in order to contribute a cleaved or in order to actually estimate a cleaved or uncleaved molecule. The second is that the informative reads that we can use based on the information we learned from our simulations are reads that fall right over the cleavage site or reads that are informative about the total number of molecules that were even transcribed. And so we walk back 1 fragment length away from the cleavage site to estimate this, and then use the difference between these two to arrive at a fraction of cleaved molecules. And finally, that polymerases can contribute reads to our uncleaved or cleaved fractions all the way through the read-through region and dependent on the labeling time, assuming that these polymerases are uniformly distributed across all of these regions. Okay, so we wanted to test our model, so we actually increased our simulations and simulated a number of different parameters across insert sizes, read-through regions, labeling time points, half-lives, read lengths, and many other parameters. And so what I'm showing you here is the correlation between our in silico simulated half-lives that are input into our simulations and the half-lives estimated by our actual model. And what you see is a really, really good correlation, a Pearson correlation of 0.99 between our simulated and our estimated half-lives. So this gives us a lot of confidence that we can move forward with this model to actually start asking about the biology of what underlies rates of 3' end cleavage. So, this shows you that the proportion of uncleaved reads decreases across time, which again is just a really good sanity check to make sure that our data does indeed show us what we think it is. And we're able to calculate cleavage, 3' end cleavage half-lives for about 7,700 annotated cleavage sites in 6,000 Drosophila melanogaster genes that are expressed in S2 cells. The median half-life is about 1.27 minutes. And so that's faster than the rate of splicing, but it's still quite variable across different 3' end sites in the Drosophila genome. So the last thing I want to tell you about is how we actually started to ask questions about what underlies variability in the rates of cleavage. And so cleavage is a fairly complicated process where it involves recognition of a polyadenylation site by this polyadenylation complex that can be recruited and alternatively recruited by a number of different adjacent and more distal sequence elements and molecular markers such as epigenetic markers. So one of the primary sequence that actually underlies this is the actual polyadenylation sequence itself, which has to be present, we think, and is the main thing that's actually recognized in order for cleavage to happen. So the first thing we wanted to ask is is there anything special about or different about that polyadenylation sequence across sites with a range of different half-lives? So we first actually located what's the likely polyadenylation sequence for each of these transcription end sites. And so what I'm showing you here are the 11 known sequences involved in these polyadenylation sites. And 2 of them, this AAU-AAA and AUU-AAA, are actually thought to be present in greater than 90% of cleavage sites. And this cleavage, this polyadenylation site is thought to occur between 10 to 30 nucleotides upstream of the actual position that's cleaved in order for the poly-A tail to be added. So we scanned in the 200 nucleotides upstream of our cleavage sites, and we located the strongest match, or the match to any of these 11 k-mers, and located where the nearest k-mer was to that cleavage site. Very reassuringly, we see a huge enrichment of these k-mers right within this 10 to 30 nucleotide range, and we see that the vast majority of them are actually these two major polyadenylation sequences. When we ask if there's a relationship between the position of the closest poly-A site and the half-life of 3' end sequence cleavage, we don't see a significant correlation at all. However, when we asked whether or not there's any k-mer that's actually enriched, we see this one of these major k-mers and another one down here pop up in the 10% fastest cleaved regions. So we were wondering what sequence feature might actually underlie this, and why do we actually see that this is correlated in some fashion? And so we asked it in a slightly different fashion. Instead of asking about the actual sequence, we asked about the number of sites that are actually present in that 200 base pair region upstream. And so what we see is that more sites, more individual k-mers that are present upstream of the site of cleavage leads to a faster cleavage half-life, where sites that actually, and a minority of sites that don't have any of these polyadenylation k-mers present actually have very slow cleavage. They still are cleaved, but cleaved much, much slower than sites that have 1 to 2 k-mers, 3 to 4 k-mers, or 5 to 10 k-mers upstream. So this implies that maybe the density of sequence elements upstream of the polyadenylation sequence is actually leading to more recruitment of these polyadenylation complexes, and therefore faster cleavage of the given site. Finally, we asked about whether or not we could actually understand something about alternative polyadenylation sites and whether they are actually driven by different cleavage rates. So we broke down our cleavage sites into 3 categories: those occurring from single last exon genes, where a gene only has 1 possible cleavage site; alternative last exon genes, where a gene has 1 or 2 or more last exons that are entirely independent, such that the cleavage site occurs in independent exons; or tandem 3' UTR genes, where the cleavage site occurs in the same exon, but there can be multiple different cleavage sites present in that same last exon. When we look at the aggregate rate of cleavage across these 3 different categories, we see that genes with a single transcription end site are cleaved the fastest, while genes with alternative choices of cleavage sites to be used are cleaved at a slower rate than this kind of group of default only one cleavage site to be used. We can probe into this a little bit further and ask more about the positioning of these alternative sites. And what we see is that for both alternative last exons and for tandem 3' UTRs, the distal or downstream most cleavage site tends to look the most similar to the single last exon, the distribution of these single last exon half-lives, while the proximal or upstream most cleavage site tends to be cleaved at a slower rate than any other cleavage sites in that actual gene. And so what this is actually potentially indicating is that there might be competition between other mechanisms or other factors, including splicing, that influence the usage of this proximal site. potentially that polymerase or the polyanylation machinery might actually recognize that there's another downstream site left to be transcribed, and therefore slow down the actual cleavage at this proximal or upstream site. So we're doing a few more experiments and also analyses to try to understand what might be underlying this. Okay. So in the last kind of 3 minutes, I want to tell you a little bit about the other part of the work that we're doing, and that is that we're asking about the balance between the speed at which mRNA is actually processed, which I've just told you about for most of my talk, and how accurately these mRNAs can actually be processed. And so similar to how many other fields think about this, we actually think about there being a trade-off between how fast you can actually produce a final product, in this case mRNA, and how well you can produce it. Where if you really go for speed, you might actually compromise on the ability to do something accurately, and vice versa, that if you want to do something really, really well, you might actually have to take your time to do it rather than do it fast. So we think something similar is going on with mRNA processing. And what we want to do is we actually want to measure, well, what is the accuracy or fidelity of mRNA processing reactions? So the concept of fidelity, or and in particular error rate, has been measured extensively in many other processes, including DNA replication, transcription, and translation. But much less is known about the fidelity or the proportion of mistakes that are made in the context of actually splicing or cleaving an mRNA molecule. Let me go straight to, okay. So one thing that we do know from previous literature, including work that I was involved in, is that there is actually a relatively higher degree of erroneous or noisy splicing than was previously thought. And so what's being plotted on the right-hand side here is that across intron lengths in the human genome, what's the mean fraction of unconserved splice sites that are present. And so the longer the intron, the more that you see these erroneous splice sites that are not actually matched to a known canonical splice site sequence, but rather occur somewhere in the middle of a gene. So while this was seen to be common, it was actually quite hard to find with traditional RNA sequencing data sets. And that's because even though this data set used 1.2 billion polyadenylated RNA sequencing reads, because they were polyadenylated RNA sequencing reads, they only represented mature mRNA. And the thought is that these mistakes in splicing or noise in splicing might actually be degraded quite quickly by a number of mechanisms, including NMD, in order to prevent them from contributing to error in protein production and harming the cell. So what we realized is that our nascent 4SU RNA sequencing might actually get around this limitation of only looking at mature molecules and allow us to study non-canonical splicing reactions. And so one way in which we did this in flies, using the same data that I just told you about for kinetics, is to actually look at the rate, the presence of a non-canonical splicing reaction called recursive splicing. So canonical splicing occurs, as I mentioned, where an intron lariat is taken out as one piece. But recursive splicing is a situation in which there's a juxtaposed 3', 5' splice site, and this intron is actually taken out as multiple pieces. And what this results in is a very interesting difference in the mechanism that's not seen in the final product. Because as you can see in this example, the final product of both canonical and recursive splicing looks the same. It looks like two exons that are spliced together. But the usage of this stepwise splicing reaction actually has a lot of implications for the mechanisms of how efficiently or accurately splicing might occur. Previously, using again 10 billion RNA sequencing reads from Drosophila, only 130 recursively spliced introns were actually discovered. But we used our nascent RNA sequencing data and 3 new machine learning methods to actually develop new techniques, new approaches to identify recursively spliced introns. And what we were able to do is we were able to actually increase the number of recursively spliced introns and sites by fourfold, identifying about 500 recursively spliced introns in the Drosophila genome. We see that longer introns tend to have recursive splice sites. Longer recursively spliced introns tend to have more recursive splice sites that are not randomly distributed, but rather fairly evenly distributed across these introns. And these sites have very strong and very conserved motifs, indicating that they might actually be under quite strong selective pressures. So we asked whether or not recursive splicing might actually result in an increase in the speed of splicing these introns, since we just looked at the rate of splicing. What we found was that recursive splicing doesn't seem to correlate at all with an increased speed in splicing out these introns, but rather this piecemeal process of splicing out an intron actually resulted in a decrease in noisy splicing of these introns, where we think that having this recursive site present in the middle of an intron and evenly spaced across longer introns might actually act as a beacon to pull splicing machinery and recruit splicing machinery to it, rather than hitting a random site that might be a cryptic splice site occurring in a very long intron. And so what I'm showing here is that recursive introns in yellow have a much lower, significantly lower proportion of non-canonical junction reads relative to non-recursive long introns in the Drosophila genome. Okay, so that data is actually published. And but with that, I'd like to wrap up by telling you that that was a piece of the 3 stories that are related to each other, talking about the rate and accuracy of splicing. We're now following up, as I mentioned in a few places, with other methods and other applications for studying the kinetics of splicing and applications to study the accuracy of splicing, and specifically get into the mechanisms and therapeutic consequences of mistakes or noise in splicing. And with that, I'd like to thank everybody in my lab, many of our collaborators and funding sources, and end with an advertisement that we are very much recruiting students and postdocs. And so this is an ad for doing a postdoc in my lab, where we're interested in recruiting people who are both experimental and computational, and really want people who are collaborative and excited about the work we're doing, RNA biology and computational biology. So I'll leave this up here, and please feel free to email me if you're interested in any way. Thank you. So I'm happy to take questions if we have time. I know I might have run over a little bit.
I put some notes in the chat with 2 questions from myself.
Great.
Well,
Gloria, go for it.
Well,
sure.
First of all, I'd like to thank you so much for the marvelous, very interesting and very exciting seminar. It was wonderful. Thank you so much. We have some manifestations in the chat, but I cannot access now. I don't know what's happening.
Yeah, I'm trying to find the chat myself.
Yeah. Here, I found it.
Okay.
First, Miguel, would you like to ask to her?
Oh yeah, I was taking notice there because I would like to ask you. I was writing those questions during the Intron lifetime, right? Sure. So I was wondering if you could analyze separately early replicating or late replicating genes, if you have a map of those, or highly acetylated, deeply acetylated, like in categories of chromatin structure, to see if there is some variation, some effect. Right.
Yeah, we did a little bit of analysis and correlating to histone marks, but I think there's a lot more we can do. So yeah, we haven't looked at it, but it's a great question to look at the surrounding chromatin environment, DNA methylation environment, histone marks, nucleosome positioning, and also modifications in RNA itself. And that's another area that I think would be very interested in. And there's more and more data, especially in Drosophila, to look at that. So I would love a student to come and join us and do that. I think it's a great idea.
Oh, yeah. And I noticed that your distributions, they are quite close to normal distribution, Gaussian distribution, right? For all your data. And I would like to know if you have some outliers or distant outliers. Maybe those genes would be high interested to study specifically.
Yeah, that's a very great, but I didn't point that out, but you're entirely right. We do have a number of outliers, and for a few reasons. So one, I didn't go into either for the splicing rates or the cleavage rates, but we actually used our simulations to understand what our limits are. So where do we not have any confidence in our rates? And it turns out that the numbers are different for both splicing and cleavage, but for splicing, this is published, and go look at the numbers. But the gist of it is that we don't have very much power for genes that are very, very quickly spliced, or genes that are very, very slowly spliced. And that's because of the limitations of the time points that we use. So one thing that we're actually working on in our lab right now is to reduce the time. So to do shorter time points and actually ask about, well, what's the rate of those genes? Because we've been focused on genome-wide methods, we haven't really gone after those outlier genes, but we do know that they are extremely fast or extremely slow spliced genes, and that would be very interesting.
Did you try to do some enrichment, even though, to see if they are from a particular class of genes?
Yeah. So I'm trying to remember for splicing specifically what we found. So there's a little bit of a caveat, and that is, I didn't go into this, but there's some mechanistic insights we realized, and that is that, so first I'll say that there's actually a correlation between the rates of splicing across different introns in the same gene. So different introns in the same gene tend to be spliced with more similar rates, or lower standard deviations after controlling for a number of things than introns from different genes, except for the very first intron in the gene. So the very first intron is a very, very long intron in most eukaryotic genomes, and definitely in Drosophila. And so in order to see that correlation, you have to take out the first intron. So it seems like the very slowly spliced introns are first introns. So they don't tend to necessarily occur in any specific types of genes. They just generally are this class. However, some of the quickest spliced introns are, I'm actually totally blanking on what the gene ontology category was. I think there was one gene ontology category that was significant, and it we didn't really quite understand it because it was very weakly significant. Okay. But you should definitely go look it up, and I'm happy to discuss it more. I need to refresh myself on it. The longer ones, I think, were interesting, and were actually one of the reasons that we started looking at recursive splicing, because we wondered whether or not recursive splicing was playing a role in these long introns. And yes, it is. However, it doesn't seem to be playing a role in the speed of the splicing of them. It's just that recursive splicing does tend to occur more often in these very long introns. It's a great question.
Great. We have other questions in the chat. I will read for you, Athma. Sure. Is from João Paulo. Miguel, sorry.
No, if João Paulo wants to ask.
If João Paulo wants to ask some for himself, or
yes,
okay, great.
I'm here.
Oh, great.
Thank you, Professor Gloria. Thank you, Dr. Athma, for the presentation. It was really fascinating. My question is about antisense RNAs and antisense transcription. Okay. If you ever take a look at the possibility of these two phenomena influencing in the splicing rate.
Do you mean antisense transcription from the promoter region? Or?
Yeah, no, from internal promoters in the coding regions.
I see. We have not looked at that. We should. We haven't. I mean, I can tell you that this was all stranded RNA sequencing data. So we did, only use stranded reads. So I think we're not confounded by that. But we haven't looked at that. We did look at We did look at whether or not the presence of a small RNA being processed out of one of the introns influenced the splicing rates. And I don't actually know in Drosophila whether or not those tend to be antisense or not, but I think so. And we don't see any relationship between that, but we haven't looked at non-small RNA processing. So it could be very interesting to look at that for sure.
Great.
Thank you.
Thank you. There is another question from Isabella. Isabella, would you like to ask your question?
Yes, I want to ask my question.
Yeah, great.
Very, very interesting talk. Like me and I'm Gloria's student. We also work with splicing.
Great.
And I have a question. What is the depth, the depth for the transcriptome that you were needed to reach the splicing time results, and also the length of the reads? Because I imagine that needs to be a very deep transcriptome.
So yeah, so for this was a 50 million reads, 50 million paired end reads for across 3 replicates. However, that's Drosophila, which is a much shorter genome. So that's 50 million reads for a 300 megabase genome. So that's what we developed the method with. However, I think you can actually cut that down quite a bit for Drosophila. We definitely had way more than we needed. And so in the supplement of the paper, buried in there somewhere, we subsample the reads and show what the read depth that is necessary to be able to get the same results is. And I don't remember right now what the exact number for Drosophila is. Your second question was about read length. That's a good question. So in this particular case, our read length was actually 50 nucleotides long. But again, they're paired end reads. So we actually took advantage of the 2 pairs, and we could infer whether or not the insert size between the 2 pairs, given a known insert size distribution, whether or not that pair of reads came from a spliced or unspliced molecule. Longer reads would be great. Yes, I think it would hugely increase our power because it would allow us to capture a lot more of these junction reads for sure. And so in our lab now, we only sequence pretty much 100 or 150 base pair paired end reads, especially from mammalian genomes.
Thanks a lot. I was having that thought because when I look at alternative splicing in normal bulk transcriptomic data sets, I need to, for human genome, I need at least 20 million, 30 million reads to have a good number of results.
Yeah, for sure. And so for the human genome, I would estimate, we haven't done these calculations because we're still generating a lot of these data. For the human genome, I would estimate you need at least 100 million reads to do a splicing rate calculation. For the Drosophila genome, I don't think that's necessary. We can go quite but yeah, for the human genome, you need quite a bit. So
thanks a lot, Doctor.
Great. Athma, I have many questions I would like to ask you, but I'll concentrate in some few ones. Sure. Concerning the rate of the splicing along the RNA, you told to Miguel about the first exon, first intron as being the bigger one.
Yeah.
But I have seen in other organisms that some long introns happen in the end of the transcript.
Yes, they do.
What happens with the rate of splicing with these long introns in the end?
Right. That's a great question. So let me actually see if I have that slide here. I think I might actually have a hidden slide for that. So one thing that I didn't talk about actually is we went into here. I think I can show this. Sorry. So one thing that we go into in the paper, and I just didn't have time to talk about today, is the difference in intron versus exon definition. So you might be familiar with this. This is like an idea that, introns, splice sites can either be recognized where you pair across the intron, or they can be recognized where you pair across the exon. So that defines the exon first and then pair across the 2 exons later. So what we were curious about is whether or not that global intron length versus splicing rate distribution might actually be oversimplifying it, because we're in the Drosophila genome in particular. There, I have the slide. So in the Drosophila genome in particular, there's a mix of intron and exon defined introns. And that's actually not common. In mammalian genomes, they're almost all exon defined. In yeast, they're almost all intron defined. And it seems to be based on the relative length of introns and exons. And so Drosophila, and I think C. elegans as well, are pretty unique in this combined distribution. So we decided to take advantage of that and actually ask about, well, what happens with intron versus exon defined intron. So we devised a metric that, you know, is a little crude, but the best we could do on actually trying to define which introns are intron versus exon defined. And what we see is when we actually break up our introns into intron versus exon defined introns, that global picture that I was telling you about becomes a little bit more complicated, because for intron defined introns, you actually see that this diffusion model seems to be true. And that is that for intron defined introns, again, where you're pairing across the introns, so it seems like the intron length should matter. The longer the exon, the slower the splicing. So, you know, the more the distance between the sites, the slower it takes to actually splice out an intron. For exon-defined introns, where exon length probably matters a bit more, you don't see that. What you actually see is that is this pattern where you tend to see that the longer the intron, the faster spliced out it is. So a lot of this is taking first introns out because, again, we did various things where we tried to analyze first versus non-first, and so. Most of what you're talking about, introns that are very, very long, but not the first intron, fall into this category right here. So it seems like they're actually spliced out faster than other introns, which is a little strange, but interesting. We don't quite know why this is.
Very, very interesting. And that depends on the elements, like the intronic sequence splicing silencers or intronic splicing enhancers, and what kind of RNA binding proteins are bound to that element. You didn't comment about the ISS, ESS.
So we have some K-mer analysis. The major things that we found that are associated, and this is one of the reasons I actually skipped this slide because I would have to explain intron versus exon defined introns, but these are so when we do a regression analysis and actually ask about what are the things that contribute and what are their relative importance to the rate of splicing across these two different categories, intron versus exon defined introns, we actually see that there's a number of things. There's no specific K-mer or specific RNA binding protein that we could identify that was associated, but AU content definitely matters quite a bit, which, you know, is obviously associated with a lot of RNA binding proteins and stability and things like that. The length of the first intron, so these are again non-first introns that we're modeling. The length of the first intron, speed of splicing. Having an enhancer in the first intron actually matters. There's some more analysis we did with splicing enhancers, splicing silencers, and there's a number of things that matter. Obviously, the strength of the splice sites matters quite a bit as well, but in opposite ways, right? So the 3' splice site matters more for exon definition, but the 5' splice site matters more for intron definition. So I think this is actually really fascinating. And one of the reasons I think this is fascinating is because we always think of Drosophila as being a really great model for mammalian splicing. And obviously it's been used for many years and it's extremely conserved, all the factors and the sites and everything. And I think that's kind of really underselling the story because obviously there's some really interesting mechanistic differences going on. So, I mean, the first is this idea that, you know, Drosophila has two different sets of introns that the mammalian genome doesn't really have. And the second is that as much as people have searched, the mammalian genome has very, very long introns and doesn't seem to have as much recursive splicing. So now there's two things that are indicating there's something going on with the splicing mechanisms and how they're how evolution is acting on them, how the sites are being selected, the architecture of the genes that's inherently different between the two species, and is worth studying. So, yeah.
Great. I have another question.
Can I ask a question, Claudia?
I didn't see there was a question in the chat. So
Somebody else just said.
well, it was not. It was not in the chat.
Yeah.
Okay.
So, yeah. Hi, Dr. Athma. Great talk. So I was wondering if
sorry, who is asking?
Eddie.
It's me, Eddie.
Okay.
Yeah,
Eddie.
your ex-student.
Eddie. Oh God. Nice to see you. Show yourself.
Good to see you.
Where are you from today, Eddie?
Sorry?
Where are you from? In Brazil?
No.
From New York.
New York.
In Cornell. I miss you. I miss you so much.
That's clear. Yeah. So I do have a question. Actually, it's two questions, but they're related. Have you looked what is the correlation between like your splicing and cleavage estimates and RNA velocity derived from single cell experiments? And related to that is how does because I understood from your experiments, you're analyzing these rates from bulk experiments, right?
Mm-hmm. Yeah.
So even though I guess you starve the cells and like have them like all on the same time, there's still a factor of the bulk, especially if you consider like tissue heterogeneity, since like some tissues have splicing and cleavage size preference. So how does that plays into your model?
Mm-hmm. Sure. So, so I can tackle the first question first. We're in the process of doing that. Literally, now. I think the rates are running right now to compare splicing and cleavage rates. So email me in a week. We'll hopefully have that. And we haven't really done anything with RNA velocity because we have not compared to single cell data at all. And one of the reasons is what relates to your second question, and that is that these are all cell lines, which means that as much as the cell line is homogeneous, these are fairly homogeneous. And so the variability is more related to polyploidy in the cell lines than cell type composition, because these are clonal cell lines that were growing up. So we don't think that any cell type specific or tissue type specific differences in alternative splicing should impact these results, because we're not doing it on a heterogeneous population of cells. It would be interesting someday to actually look at that, but that's a little bit in the future. So.
Great.
Thank you.
Thank you.
Thank you, Eddie. There is another question in the chat by Thomas. Thomas, please.
Hi, Dr. Pai. Thank you for your talk. I'm also a student of Gloria, so we are a gang here. So my question is, since we have been working with bulk RNA-seq and with the gene code annotation, state-of-the-art annotation for transcripts that are not protein coding for protein coding genes, we are dealing with a lot of doubts about how to interpret our results from bulk RNA.
Right.
So my question is, if we are actually annotating too many novel transcripts, and if the gene code annotation is kind of inflated due to the fact that we're not analyzing the intermediates of RNA processing, are these transcripts actually useful? Are they going to be functionally relevant for the models that we're studying, or how can we be aware of that?
Yeah, that's a great question. So I would argue that analyzing intermediates will actually even more increase the number of alternative isoforms that we see. And we see this actually in projects that are ongoing in the lab that I didn't have a chance to talk about. Some of the slides I skipped in the interest of time, actually. And that is that we can identify a ton of unannotated, and now I'm talking about human data. So in Drosophila we can as well, but in human data, we can identify a ton of unannotated or even non-canonical splice site usage at fairly high levels in ForestView, so nascent RNA sequencing data. So if you were to include those in a gen code annotation, you'd have maybe double the amount of transcripts. So my inclination is that, yes, you're entirely right that they're already inflated. But I think that in my mind, the reason that they're inflated is because the vast majority of a lot of these alternative isoforms occur at a low level. If they, if they don't trigger an NMD isoform, or if they're not caught by some kinetic proofreading mechanism, they're relatively stable. They occur and they last, and as long as they don't have a gain of function effect, they spend their lifetime in a cell and they occur for whatever reason. And so therefore, I would argue, like I usually lean away from identifying novel isoforms in any data set, because I don't know that we need to anymore, right? If there's a specific gene that you're interested in, then yes, for sure, right? I mean, then you have to go in and characterize them. But I think on a genome-wide level, we've probably reached saturation quite a long time ago in terms of the functional isoforms in general, not context specific. So, I mean, I think one of the things that we're interested in and happy to collaborate on is whether or not we can identify whether there are specific sequences or splice site contexts in which there's more noise likely to happen, right? And if you start to understand, what are the mistakes that happen in splicing using these intermediates, and then use that to understand what mistakes are more likely to be long-lived versus short-lived, you might actually start to go back and look at these, canonical splice site alternative isoforms that are low frequency and ask, are those actually just mistakes, right? Are those just long-lived mistakes that are happening in the genome? So that's my viewpoint, which might be a little radical from the perspective of this, but it comes really from this efficiency standpoint of splicing is not that efficient, I don't think, right? It's not as efficient as the textbooks would lead us to believe. And so, RNA sequencing has been a boon for the field, but also led to probably a lot of uncovering of things that, are a result of that slight lack of efficiency. So yeah, that's a long-winded answer to say, yes, I think it's entirely inflated. I don't know numbers, right? It's impossible to say. And I think it's almost impossible to say, in what cell type context could a particular isoform be regulated versus not. That's really hard. But.
Great, thank you.
Thank you.
Well, I'd like to ask the last question I have here.
I have one more.
Well,
no, but after you.
Sorry, it was very, very interesting talk, Athma. That's why I was so excited.
Go for it. I will ask you one after you.
Yeah. Sure.
Okay. Thank you, Miguel. Do you think that alternative polyadenylation signals or sequences can be connected to the TSS, the start site that is being chosen in a moment? Well, because you didn't comment about different starting sites and how they influence the splicing, but they can influence the polyadenylation start signal recognition.
Yeah, no, definitely, 100%. I think they definitely can be influenced in ways that I don't understand yet. But I think that the correlations are definitely there. I think that it's very clear in a lot of data sets that I've looked at or we've been working with. So yeah, I mean, that's one of the things we'd love to analyze, especially once we have, all of our rates in place. We have our splicing rates, we have our cleavage rates. We're also, I mean, I should mention that we did a lot of this work in Drosophila, which is nice because we can get really, really high coverage, so we can parse back and say, what do we need? But we're actually now doing this all in mammalian cells as well. And so once we have that, and then we also have transcription site identification and transcription elongation measurements, I think that to me would be a great data set to actually ask about the mechanisms that underlie those connections that you're talking about. So yeah, I think it's really, really fascinating to study that. And I definitely think there are a lot of connections that we're building towards. We're not quite there yet, unfortunately, but yeah.
Okay, thank you so much. Miguel.
Yeah, I know that you are looking at the canonical splicing site, but I wonder if it's possible to deeply look at the splicings that would provide a subset that would go to nonsense mediated decay, specifically, if they are kind of too quickly processed or slowly or whatever.
Yeah. So I'll share one other slide that I unfortunately had to skip, and this is actually something we're we're actively doing right now. So as I mentioned, I mean, I mentioned this in the context of recursive splicing in Drosophila, but we're doing this in mammalian cells now too. So this is actually not our data. This is data from a neuronal cell line with 30 minutes of 4SU labeling. And what you can actually do here is you can, this is data analyzed by my postdoc, Erich Kocher, where what we've done in the middle here is we've actually kind of squashed all the annotated isoforms into one, just to give you a sense of where are the annotated splice sites. And then we scanned this entire gene. It's a gene called SLC6A2. This is a neuronal GABAergic neuron gene. And we scanned the gene body and identified all cryptic 5' and 3' splice sites. So anything that loosely had a splicing signal, right? Not very strong, obviously, just anything that could be kind of within the range of a splicing signal, and those are marked here. And so one thing that's really interesting is that if you look at all of these, right, a lot of them cause NMD, but the question is, are they used, right? Like, are they just there and do they not get used? Do they get used? And so now what we can do is we can put on top of it the junction reads from this 4SU sequencing experiment. Keep in mind, this is 30 minutes. So we actually think a fair number of things that could be triggered by NMD could have actually already been degraded. But what you can see in black is the annotated junctions. So these are junctions that match annotated splice sites. So those are most likely to be things that have been seen before and are more stable. And then in red are the ones that are unannotated. And what's really interesting is now you can use these data to actually start parsing, which of these sites are actually used. And these sites don't appear, the red ones don't appear in steady state data from the same cell line. So yes, we think that these are essentially targeted by NMD. And we can do this genome-wide, and what we're starting to do is develop a model to actually ask about, can we identify these sites and then determine, why are they triggering NMD or triggering something else, right? So it turns out they can also be triggering exosome mediated degradation. They could be triggering a non-stop decay, NSD. They could be targeted by XRNs, either one or two. And so we're of the mindset and doing some analyses to try to determine that using these data sets that will give us some indication for what's happening and then what's happening to those RNAs that are cryptic or mistakes. But yeah, that's all still in progress. Yeah.
Okay, Gloria.
I think you're muted, Gloria. If you're
muted, Gloria.
Sorry.
Yeah.
Well, I don't think we have more questions. And then I'd like to thank again for the amazing presentation.
Thank you.
Amazing data. Congratulations for your work. It was really wonderful to have the seminar today. And you can see by the manifestations and questions that we're asked. Thank you so much very much.
Questions too. These were really, really great questions. And so I think, some exciting thoughts here.
Okay, great. Thank you, Dr.
Great.
Athma.
Thank you so much.
Thanks a lot, Athma.
Thank you. Thanks a lot. Bye-bye.